All right, so this video or videos, depending on how long it is going to cover topics 4.1 through 4.4 of the AP College Board for AP Bio. So these slides are actually going to correspond to sections 5.5 and 5.6 of your textbook. So those are the chapters and sections that you can find this information in, but it does cover about 4 topics according to the College Board. So this video is about cell membranes and signaling. So cell communication, how do cells respond to environmental signals, and how are those signals transduced within the cell so that the cell can respond to the environment. So I have a quick little picture here that shows you this. We have a sending cell sending ligands to a receptor of a target cell. That's going to trigger a response or a signal cascade within the cell. So we have this cascade, this transduction pathway of the signal bounced around like a baton from place to place to ultimately wind up in a response inside of the cell, right? That's basically the overview of this. So cell-to-cell communication is essential for both multicellular and unicellular organisms. So biologists have discovered some universal mechanisms of cellular regulation. So cells most often communicate with each other via chemical signals or ligands, as we'll typically refer to them as. We have an example, the fight or flight response that's triggered by a signaling molecule called epinephrine. So cell signaling, cells can process information from their environment. Signals can include physical stimuli, heat or light, or chemicals known as ligands. So the cell must have a receptor for the signal in order to respond. And then we have the signal transduction pathway, which I was talking about for a second ago. And that's initiated after receptor activation by signal and is a sequence of events that lead to a cellular response. So that's going to happen in about 3 general steps that you're going to have to know. The first one is going to be reception. The signal is received. That signal is transduced within the cell, and that's going to lead to some response. And there are a couple of different types of responses it can have. So if we look at this here, the chemical signal is detected when the molecule binds to the receptor protein located at the cell's surface or inside the cell. So we have the signaling molecule that's in the shape of like a teardrop. It's going to fit into the receptor that has a cutout that would fit a teardrop. So that's reception. It's going to convert that signal to a form that can bring about a specific cellular response. So we have relay molecules in a signal transduction pathway. So that's transduction. That happens because the signaling molecule attached to the receptor. That transduced signal is going to trigger a specific cellular response. So that's going to be activation of a cellular response. And again, we'll get into that, more specifics of that in a little bit. So the signal transduction pathway is going to be when a received signal is converted to a specific cellular response in a series of steps. So the signal transduction pathway. So in a multicellular animal, cells are exposed to many chemical signals, and we have different types here that you're going to have to know. So you have autocrine signals. Those affect the same cells that release them. So I have this one right here. The cell is releasing little ligands, and those little ligands are getting received by its own receptors. You have paracrine signals. Those are signals that are going to diffuse to and affect nearby cells, just like what's happening here. And then you have juxtacrine signals. Those are going to require direct contact between the signaling and responding cell, just like we have here. They are directly contacting each other. And then you have hormones, which can travel to distant cells from wherever they're produced. So a signal molecule can act on the cell that produces it, on a nearby cell, or be transported by the organism's circulatory system to a distant target cell. And that's going to be more like hormones and stuff like epinephrine and whatnot. So I got those 3 steps. Step 1, signal binds to the receptor. Step 2, the binding induces the signal transduction. And step 3, the cell responds accordingly. So let's focus on step 1, signal binds to the receptor. So how can reception happen? So only cells with the necessary receptors can respond to a signal. The target cell must be able to sense it and respond to it. So a signal transduction pathway involves a signal, a receptor, and a response. So we have the signaling molecule getting picked up by the receptor, and then ultimately we get a response. And that can either be a short-term response or a long-term response. And we'll talk about those. So receptors can be classified by their location. They can be intracellular receptors, and that means they're located inside a cell. Typically small nonpolar ligands and can diffuse across the membrane are going to be the ones that can get to those receptors. Then there are membrane receptors and located on the cell surface. So large or polar ligands that cannot diffuse through the membrane are going to be the ones that can signal those membrane receptors, or typically that's what's going to be, those receptors are going to use as their ligands, are ones that are like this. So ligands. Ligand receptor binding is non-covalent and reversible. Non-covalent, reversible. So reversible binding is going to allow the cells to stop responding to a signal after the appropriate response has occurred, and that's definitely a good thing. So inhibitors or antagonists can bind in place of the normal ligand. So caffeine, for example, binds to the receptors in the brain, preventing the binding by the normal ligands that would make you sense that you're tired, right? So caffeine is going to prevent your brain from sensing or receiving the signal that it's actually tired. So there are three major types of cell surface transmembrane receptors. There are ion channel receptors, protein kinase receptors, and G protein coupled receptors. First one, ion channel receptors. These are ligand-gated ion channels. So once the signal binds, the gate will open or close, and that's going to allow or block the flow of specific ions. So this can be voltage gated, so that's going to be controlled by electrical signals instead of ligands, and that's going to be a critical thing in the nervous system, which we kind of talked about already this year. Once that binds, it's going to change shape. So acetylcholine receptors, for example, on skeletal muscle cells bind acetylcholine to open the channel and allow sodium to diffuse into the cell. And we've talked about this one already, but we didn't name it. But that's a ligand-gated ion channel. We have protein kinase receptors. Protein kinase receptors also change shape when a ligand binds. So the new shape exposes or activates a cytoplasmic domain. So on the cytoplasm, the internal side, that has protein kinase activity, which means it's going to modify proteins by adding that phosphate group to it. So not all protein kinases are actually receptors. So that's just like a little side fact, okay? Membrane receptors that attach phosphates to tyrosines or other proteins. So kinase is an enzyme that catalyzes the transfer of a phosphate group. And G protein linked receptors, a ligand will bind on the surface, which is going to expose a site on the cytoplasmic side that binds to a mobile membrane protein, which is a G protein. So the G protein receptor is partially inserted into the lipid bilayer, and it'll have a partially exposed side on the cytoplasmic surface, so on the inside of the cell. Many G proteins have 3 subunits that can bind 3 different molecules. They can bind the receptor, that's one of those subunits is the receptor. They have a GDP and GTP used for energy transfer, and they have an effector protein that causes an effect in the cell. So the activated G protein linked receptor exchanges a GDP nucleotide bound to the G protein for a higher energy GTP. So we're going from GDP to GTP. The activated G protein will activate the effector protein, leading to signal amplification. So this G protein we got going on here is an intermediary between the receptor and the effector protein. So if we look at this here, we have a signal. It's going to bind to the G protein linked receptor. So it's binding to the receptor, and we say this receptor is linked to a G protein because it is. We have an inactive G protein that's linked to this receptor, so that's why it's called a G protein linked receptor. So we have this inactive G protein at this state, and we have an inactive effector protein. But once this ligand binds, we're now going to change that GDP to GTP, and we're going to activate that G protein. So that activated G protein, part of it, can become mobile, move to that effector protein, trade its phosphate to it, right? Break that phosphate off and power the activation of this effector protein so that this protein can create something create some product so that the product can allow for some cellular response. So this is this whole G protein linked receptor business. That's probably one of the more complicated ones to understand, but it's pretty straightforward, right? So the second step to this, so those were the different types of receptors. The second step to this is binding is going to induce a signal transduction pathway. Signal transduction, right, is going to happen after reception. That pathway is going to lead to a cellular response. So signaling can initiate a cascade of protein interactions. That initial signal is amplified and distributed to cause different responses, ultimately leading to changes in cell function, so that the cell can do something and respond to whatever signal it got. So how to turn a protein on or off during transduction. That has a lot to do with phosphorylation, which we've talked about this already. Phosphorylation is one of the most common ways to alter protein activity. So what's going to alter those proteins? Going to be protein kinases, which are enzymes. That's going to transfer the phosphate from ATP to a protein, a process called phosphorylation. And then you have protein phosphatases, which are going to remove the phosphates from the proteins, a process called dephosphorylation. So the phosphorylation and dephosphorylation business acts as a molecular switch, turning activities on and off, or up or down as required by the cell to help this signal transduction pathway. So we have an example of a phosphorylation cascade. So we have a signaling molecule, the ligand binds to the receptor. It's going to activate a relay molecule, which is going to go from, we have an inactive protein kinase and the activated relay molecule to an activated protein kinase. We have ATP to ADP. That's going to help this business out, power this. We have another split between ATP and ADP to activate another protein kinase. So it's basically like a passing of a baton, right? We're passing this baton from one point to the next point to eventually we end up activating some protein that's going to lead to some cellular response. So it's pretty involved. These processes are pretty involved. So what else can propagate transduction? Second messengers. So second messengers are going to be small non-protein molecules that pass along a signal initiated by the binding of a ligand. The ligand is considered the first messenger, right? So this is going to regulate target enzymes by binding to them non-covalently. So why are second messengers necessary? They help distribute the signal. They allow the cell to respond to a single membrane event with many events inside the cell. And most importantly, they amplify the signal by activating more than one enzyme target. So second messengers include cyclic AMP or CAMP. This is a derivative of ATP, so something similar to that, and that's one of the most widely used second messengers. Another one is going to be calcium ions. That's going to be widely used by cells and can be stored in the endoplasmic reticulum. And then inositol phosphates. Those are derivatives of phospholipids. So these are some important second messengers. More about CAMP, because this one's a really important one. CAMP can activate an enzyme called protein kinase A or PKA. That will enable it to phosphorylate its target and pass along the signal. PKA is found in a variety of types of cells and has different target proteins in each. This allows the same CAMP second messenger to produce different responses in different contexts, which is, a big reason why CAMP is important. So if we look at this here, we have a ligand binding to the receptor. That's going to cause some response in this dinylyl cyclase, which is an enzyme. So we're going to go from ATP to CAMP. CAMP is going to help activate this protein kinase. It's going to activate it, and then that could lead to some cellular response. But what actually activated that, right, was the CAMP itself. That's why we consider CAMP the second messenger, because it was necessary for this second messenger to be activated or to activate this protein, right? So the ligand is the first message or messenger, and then CAMP would be the second messenger in this example. All right, and the third step is the cell responds accordingly. So the response, ultimately a signal transduction pathway, leads to the regulation of one or more cellular activities. So the response can happen in two places. The response can either happen in the cytoplasm of the cell, or it can happen in the nucleus. Nuclear responses. In these, the signaling pathways can regulate synthesis of enzymes or other proteins, usually by turning genes on or off in the nucleus. So the final activated molecule and signaling pathway may function as a transcription factor. A transcription factor is going to be one that determines whether or not protein synthesis starts or stops. Remember, transcription is the first step of protein synthesis, right? So when we say something's a transcription factor, it's going to regulate which proteins are created or not created. So a response is going to end transduction. So signal transduction ends after the cell response. The enzymes convert each transducer back to its inactive precursor. The balance between regulating enzymes and signal enzymes determines the cell's ultimate response. So cells can alter the balance of enzymes in two ways. Either they can have the synthesis or breakdown of the enzyme, or they can have the activation or inhibition of the enzymes by other molecules. And there are many ways in which cells respond to environmental signals. You can have the opening of ion channels. That's going to change the balance of ion concentration between the outside and inside of the cell. That's going to result in change in electrical potential across the membrane. Remember, we talked about this. It's important for how neurons work. You can have alterations in gene expression. Genes may be switched on, upregulated, or switched off, downregulated. That's going to affect the abundance of proteins, often enzymes, and that can change cell function. And then you can have the alteration of enzyme activities. That's going to be a more rapid response than those involving change in gene expressions. If we can just turn those enzymes off with some inhibitors, right, that's going to allow for the processes and gene response to happen at a faster rate than if we try to go mess with the protein synthesis pathway. So we have different cellular responses. They have the specificity of the signal. That's really important here. The same signal molecule can trigger different responses depending on other signals and various receptor proteins or second messengers. So many responses can come from one signal. So if we have a signaling molecule, it could lead to just one response, or in this signaling molecule pathway, it's going to lead to two different responses. So the signal can also activate, inhibit, or create multiple responses from one signal.